All right, in the last section, what we were talking about was restriction enzymes and how they're used to cut up the DNA. So in this section, what we're going to do is we're going to talk about using that technology in a way to make basically a clone of the bacteria, so you can make a clone of that gene so that you can replicate it and make a lot of the product that you might be after. So we've used clone a number of different ways in the class. We talked about clones being identical cells or groups of identical cells. And another way to kind of look at it is genes, which can be copied from bacterium to bacterium or source to source, and then that can be a way to clone the genes. It might not, all of the genes in the organism might not be exactly the same. However, if you can get the gene that you're after, you can kind of refer to that as being a clone. We talked about clones in class as being genetically identical organisms, and we talked about strawberries, we talked about potatoes, we talked about apples, a variety of different types of things we are discussing in terms of cloning from an original parent with a desired characteristic, and kind of moving through some of the examples for how the cloning actually happens. Okay, so when we're talking about bacteria now, what we can do is we can talk about the genes within those plasmids, and that plasmid, of course, being an extra piece of DNA that's different from the nucleoid region, and it usually carries some genes that confer resistance to antibiotics or some other sort of harsh condition. And in this case, what we can do is we can put the genes on the plasmid and kind of allow those bacteria to replicate and produce a clone of the particular gene within that bacterium. And that bacterium can either be bacterial DNA, it can be eukaryotic DNA, it can be a variety of different sources that you can put it in there. But it's some sort of foreign DNA that isn't common to that particular bacterial cell. And a lot of times plasmids are used because they're very easy to manipulate. And a lot of the experimentation that you do can kind of be done right in the laboratory with these things. Okay? So the common method that's used for cloning is what you do is you isolate the plasmid within the E. coli cell that you're after, and then you take the DNA from the particular organism that you're going to insert it in. In this particular example, it could be the human DNA, so you're getting a gene that you're going to try to clone using the bacterium and the plasmid. And then what you do is you treat both the bacterial DNA and the eukaryotic DNA with restriction enzymes, and you typically know the cut site, so you know the type of restriction enzyme that you want to use, so it's going to produce the same types of cuts throughout all of the DNA. And then you mix the human and the plasmid fragments together to try to get them to kind of line up. And then once you've done that, by adding the DNA ligase together, now you can form those phosphodiester bonds, kind of link those strands permanently together, and now you've got the gene, in this case the human DNA, fused with the bacterial DNA, and you've got what's known as a recombinant plasmid. And it contains DNA from two different sources. And then what you can do is you can plate this out on some sort of a selective media, and then you can use that to isolate for particular types of recombinants, and there's other things that need to occur. But very generally what happens is what we've just described. You look at this bacterial cell that you have up here, and then within it you've got the plasmid, okay? And then you take the human cell and you've got the DNA fragments. After you've treated them with a restriction enzyme, you treat the plasmids with the restriction enzymes. And then what you do is you mix them together, and once you mix them together, treat them with the ligase and stuff, those genes just kind of insert themselves in numerous different types of spots. There's numerous fragments that occur, so you're going to have a whole bunch of different types of plasmids. And then it's your job to kind of go through and select for them. So it's not super easy. There's a lot of steps involved, a lot of really trial and error, so to speak, and that sort of a thing. But once you get these bacterial cells to take those plasmids back up, that's when you can start selecting for it to see if you have the actual gene inserted in the right spot. So typically what happens is they find a particular gene, and they're going to try to insert the gene of interest from the human fragment within that bacterial gene. So in this particular example, what we're using is a LAC-Z gene. So it's a gene that's going to break down lactose, okay? And so they're going to try to insert the human gene within the region of that gene. So in other words, you're going to disrupt the function of the lactose metabolizing gene in the plasmid, but that's okay. So then inside of this growth medium here, what they're going to do is they're going to put X-gal, and they're going to put ampicillin, and they're going to put some lactose. They're going to put a variety of different types of things in there. And what they're doing essentially is now they're going to look for and see if these bacterial cells are recombinants or not. So if they have the presence of the ampicillin resistance gene here, then they'll be able to grow. Okay, they'll all grow. So any of those things growing. So that's a way to select for some of the bacteria that might be present that don't contain the ampicillin resistance gene. So you can wipe those out and kind of reduce the colony numbers on the plate. Then what you're looking for is in these particular things, if the gene has not been disrupted, then the intact LAC-Z gene, as it's called, will be able to break down the X-scale that's been put into the medium. Okay, and what that does is it turns it blue. So the colonies that don't have the recombinants would be blue. So you can see here you've got a bunch of blue and white colonies on there. The blue ones are no good. Those did not get any gene inserted in like you're seeing in these three examples right here. What happened was is they might have broke and then they went right back together with no gene being inserted, so you can get rid of those. Any of the white colonies now here contain the disrupted LAC-Z gene. In other words, they have the fragment, or a fragment rather, in between the LAC-Z gene. So now that means that any of those white colonies could potentially contain the gene of interest. So then what you can do is do another series of experiments that aren't included here to figure out which one of those particular colonies that are white, which one of those particular colonies contains the gene of interest. So you can do certain things to kind of test for, what they might be producing or something like that. So if you're actually to do this in a lab, this is what it would look like. Again, the blue colonies here have the ability to break down the X-scale, which is present in the growth medium, and it turns the kind of byproduct of that reaction blue. And then all of the white colonies are considered to be recombinants. They have a fragment inserted in between that LAC-Z gene somewhere. And what that's doing is disrupting the LAC-Z gene's function, and therefore it can't break down the X-scale. However, it can grow because it has the ampicillin resistance, and it contains a fragment. So then you go through and you look at all of these white colonies and do experiments with those and try to figure out which one has the gene of interest. Okay, so obviously it takes a while, and there's a video here that describes how Herbert Boyer did it when he was working in the early 70s to try to get insulin placed within the bacterium so that he could get these bacterial cells to produce the insulin for diabetics. So it's pretty interesting, and you can just go ahead and have a watch. I'll post that underneath this particular lecture. Okay. So again, what we were talking about here, just in general, recognizing the clone. The bacteria lack a particular gene conferring resistance, say, to an antibiotic. And so when you plate them out, like we said, they're not going to grow. But the bacteria that contains that ampicillin resistance gene are going to be able to survive. And then what you're doing is you're looking for the one that contained the intact, the disrupted LAC-Z gene, and then you got to do the experiments with that. Okay. So here's a little video. This link works. Just download the presentation, follow it, and it'll show you just in general how the cloning action that I've just described for you works. So some of the problems now with expressing the DNA in bacteria is, as you know, bacteria lack the splicing machinery. Okay, they don't have any ways to take that eukaryotic DNA, splice out the introns, link together the exons, and get them to kind of express the gene. So you have to figure out with that particular eukaryotic gene where the introns and exons are, and then really how to get rid of them. Okay, so scientists have done a couple of different ways or a couple of different things to try to combat this problem. And what they use is expression vectors that have a very highly active promoter region just upstream from where that gene is. So in other words, they want to express a lot of the protein. The first thing is you have to do is you have to find a very active gene within the bacterial cell, because you want it to really always be on so that you can make a lot of product after you've gotten it in there. Okay, so those bacteria will recognize that foreign DNA and just express it as though it were their own. Okay, so this is how they get eukaryotic genes to be expressed in prokaryotic cells. The other problem that they have with this is you have to take the intronic DNA out and put the exons together. So what scientists have done to kind of get around this problem is they've created what's known as cDNA. Okay, and what the cDNA does is it only contains the exons. So what they're going to do is they're going to take the messenger RNA from the eukaryotic cell. They have to isolate that out, and then they take advantage of the fact that there are certain viruses in nature referred to as retroviruses. And what they do is they reverse the flow of the information. Instead of going DNA to RNA to protein like the central dogma of biology suggests, it goes DNA to RNA and then back to DNA. and then you can take that DNA and do a variety of things with it, and then that DNA will then follow the central dogma and go DNA to RNA to protein. So what you're going to do is you're going to take the messenger RNA and you're going to make DNA out of it. Okay, so what they do with this particular thing, got ahead of myself here, is they take the messenger RNA and they treat it with reverse transcriptase, and then what that's going to do is it's going to, as you see right here, synthesize a single strand of the DNA. Okay, and then from that single strand of DNA, you can add DNA polymerase to the mix, and then what that DNA polymerase will do is synthesize the complementary strand back the other way, and this messenger RNA will get broken down by a variety of enzymes and that sort of a thing. And then you can take the cDNA, you can treat it with restriction enzymes, get the sticky ends put on there, and then insert it into the bacterial chromosome without any of the introns there. You've just got a piece of DNA that contains only the exons, and you can get that put in. So there's a movie here, a link that you can follow, and it will explain, again, what I just explained to you as well. Okay? So now what you've done is you've overcome the problem with the expression of the gene as well as the presence of the introns. So the cDNA now gets expressed in that highly active region associated with the bacterial chromosome, and now that's a way for you to get a whole lot of product from the DNA, and then you can kind of purify it out. In the case of insulin, for instance, package it up and then deliver it to the people who need it. Okay, some of the other ways that scientists can get around the problems of putting eukaryotic genes into bacterial cells is just using yeast. What they are single-celled fungi, and they also, some of them contain plasmids, so that's a way that you can put the DNA in there. It's a eukaryotic cell, so it can process the DNA, it can get rid of the introns, it can express the exons, the genes that you want. You can take those recombinant plasmids, and then what you can do is you can combine them with the yeast and bacterial DNA, and you can get them to replicate in either type of cell. You can put it in a bacterial cell, and the bacterium will express the gene. You can put it into the yeast, and it will express the gene. So there's a variety of different ways to do it. But what I've basically given you there, though, is the basics for how it's done. Okay? So if you have any questions, make sure you ask, and we'll talk to you soon.